This is a message for the National Institutes of Health. I would first like to applaud the progress that has been made by the scientific community in researching Duchenne muscular dystrophy. This is a truly devastating disease that affects young boys around the world and unfortunately shortens their lifespans significantly. Increasing the life expectancy and mobility of these boys has been central to the research being carried out and medications being developed. From the identification of the mutated gene that causes this disease back in 1987, so much effort has been made in improving the lives of these boys, and in 2016, the first disease-modifying drug for DMD was approved by the FDA. There are many lines of research going on today for developing ways to improve the muscle strength of boys with this condition. However, I would like to bring attention to an aspect of the disease that needs more research: how it affects the brain. The brain is an incredibly complex system, and there is still so much to be learned about its development, functions, and most importantly, the roles it plays in diseases. Cognitive deficits are not uncommon in myopathies, and they are well documented in DMD. In this video, I will first talk about the rates of different neurological conditions and cognitive deficits. Then, I will go into the hypothesized mechanisms and causes of cognitive impairments associated with DMD. Finally, I will discuss current ways to improve neurological conditions associated with DMD and potential future avenues of study for treating DMD's effects on the brain. A 2019 study by Naito and Anthony reports that the risk of cognitive impairment is higher in DMD patients compared to the general population. Full-scale IQs of DMD patients are consistently one standard deviation below the population mean. DMD patients show increased rates of epilepsy, this is about 2 to 12% of patients. Internalizing disorders like depression and anxiety are reported in about 1/3 of patients. ADHD is diagnosed in 12 to 32% of patients. Between 40 and 50% of patients have a reading disability. Autism spectrum disorders show incidence rates from 4 to 37% in patients, and it is reported that 1/3 of boys present with at least 2 neurological disorders. There is said to be an existence of a DMD neuropsychiatric syndrome since prevalence rates of behavioral and neurodevelopmental disorders are so high in patients with DMD. Patients with DMD have a hard enough time in their everyday lives. Going to school with these disadvantages increases their hardships. Improving the cognitive aspect of the disease should be a focus in developing treatments for DMD. In order to develop treatments for DMD neuropsychiatric syndrome, the mechanism of what causes the disease state needs to be understood. Part of what makes studying the role of dystrophin in the brain challenging is the wide variety of dystrophin isoforms that are expressed in the brain. There are 8 dystrophin isoforms of different lengths, the most predominant form in the brain being DP71. Absence of DP71 and DP140 are associated with more severe cognitive impairment in DMD. More distal mutations, which have the capability of affecting these shorter variants, are linked to cognitive impairments. It is believed that mutations that affect these isoforms are associated with altered hippocampal transmission. Dystrophin is localized to higher brain order regions like the cerebellum, hippocampus, and cerebral cortex, especially at postsynaptic GABAergic neurons, which points to a crucial role in synaptic plasticity. Different isoforms are associated with different roles in the brain during development and throughout life, but there is not much clear evidence for how these roles are carried out. This also means that there is not much evidence for how mutations in the dystrophin protein lead to cognitive deficiencies. Other studies show functional imaging that indicates altered brain metabolism may be linked to impaired cognitive capacity. Higher order brain regions are hypometabolic in DMD patients compared to controls. Some studies have found elevated choline levels which indicate high cell membrane turnover, but others report choline deficits in DMD patients. Another molecule, transforming growth factor beta, or TGF-beta, is shown to have an important role in the developing brain with regards to cognition and neurogenesis in aged mice. It is also shown to be involved in neuronal metabolism and at abnormal levels is thought to trigger atypical metabolic pathways. Suffice it to say, no single mechanism has been elucidated. Despite the lack of understanding we have about what is causing cognitive impairment in DMD patients, treatments based on current understanding have been explored. Exon skipping is a promising treatment for DMD. It uses antisense oligonucleotides to target the dystrophin mRNA and allow for production of dystrophin in muscle cells that did not previously express dystrophin. A study by Sakaguchi and colleagues in 2009 used intracerebroventricular administration of antisense morpholino oligonucleotides to restore brain dystrophin expression in mice by skipping the mutated exon 23 of the gene. MDX mice show an abnormal defensive response to restraint that enhances a freezing reaction attributable to abnormal brain function. This response worsens with age as dystrophin expression in the brain becomes more apparent in mice. By restoring some expression of dystrophin in the brain, they were able to partially reduce the freeze response to restraint in the MDX mice postnatal day 65 and 80. It is still being investigated whether AONs can cross the blood-brain barrier, which is an added source of complication in restoring dystrophin expression in the brain. If AONs are able to cross the blood-brain barrier, this method could be useful in restoring expression of relevant dystrophin isoforms in the brain. Another possible treatment approach focuses on changes in neuronal metabolism in the DMD brain. Anti-TGF therapy is a developing research field and can target neuronal metabolism, which might serve as a link in the many pathways affected by the mutated dystrophin gene. Additionally, nutrition and physical activity can serve to improve cognitive deficits in DMD, but is not well investigated. Polyphenols show effectiveness in improving neurodegenerative disorders and are easy to implement into the diet by eating fruits and vegetables. Polyphenols in green tea and turmeric show a strong association with improving cognitive function and have neuroprotective effects. Overall, DMD is not just a disorder of the muscular system. The brain is also affected and adds to the detrimental effects of this disease. To review the topics covered in this video, I will summarize the major talking points. Studies indicate that DMD patients suffer from cognitive deficits at higher rates than the general population. How the mutated dystrophin protein results in this increased incidence of neurological conditions is not clearly understood. So far, research has uncovered links between distal mutations of the dystrophin gene and cognitive deficits. Also, metabolic changes in the DMD brain are observed, which could be another link to the cognitive impairments seen in these patients. Treatment of the DMD brain might be able to make improvements, but there is not a lot of conclusive evidence as of yet. Pharmaceutical options like use of AONs are in the beginning stages of research, but in the meantime, dietary changes by including polyphenols and exercising may have neural benefits. There is still so much to be learned about dystrophin in the brain, and it is important to conduct more research in these areas. I acknowledge that these neurological disorders are not fatal in DMD like cardiomyopathies or respiratory failures are, but there is already so much ongoing research on how to delay and prevent these outcomes. The goal of research in DMD is to improve the quality of life of those afflicted with this disease, so this should include all aspects of the disease, including how it affects the brain. With that, I implore you, the National Institutes of Health, to take action and increase funding for research in this disease, and to expand research to better include the brain in studies regarding DMD. Thank you for listening. I hope we can work together to help DMD patients around the world.